So it's my pleasure to introduce today's Wednesday EEB seminar speaker, Brandon Gaut, who's joining us from UC Irvine down at the other side of the 405. So Brandon is a professor in the Department of Ecology and Evolutionary Biology at UC Irvine. He was department chair for a number of years there, and he's been at Irvine since, I believe, 1998.
Right,
and
yeah.
Before that, he was an assistant professor at Rutgers. And prior to that, he did his postdoc with Bruce Weir at NC. And his graduate work, like many of our seminar speakers, in the UC system. So he did his PhD with Michael Clegg at UC Riverside and got his bachelor's at UC Berkeley. So he's a product of the UC system. Brandon has done a lot of influential work in using genomic technology for studying domestication and also impact of selection on domesticated species as well. He's done other work on cultural elements and now is moving into experimental evolution, as he'll be telling us about today. He has a variety of different honors, including President of the Society of Molecular Biology and Evolution, Professor of the Year in Irvine in 2008, and an outstanding professor, or won an award for being an outstanding professor in 2002. I'm sure he's outstanding in other years. So, without further ado, I'll let Brandon tell us about his work on evolution of genotype and phenotype and fitness and E. coli high temperature.
Thanks so much, Kirk. I'm really happy to be here, and I know every seminar speaker says that, but there's two reasons I'm happy to be here. The first is, today for me is what I call a 2-for-1. I'm coming here to give one lecture, but I got out of giving my lecture to freshman biology class, and here's the bonus, I'm missing a faculty meeting. So this is a win-win for me. Another reason I'm really happy to be here, and I hope you'll indulge me a little bit because I want to give you a little background, is that UCLA has had a huge impact on me, very indirectly. And the reason is that both my parents graduated from UCLA in 1957. So I spent a lot of time on the internet trying to find pictures from the 50s. This one actually is. It's Hillgard entrance. That's from 1929 when UCLA opened. But it was kind of fun to talk to them about what UCLA was like in the 50s. They actually lived down in Hawthorne. That's where they met and drove to campus. And you think traffic is bad now. It took an hour to get to campus from Hawthorne then. That's just near the airport because there was no freeway. Another thing that hasn't changed is I said, so why didn't you move and live closer to campus? Because it was too expensive, right? So we know all about Westwood in that way. One thing that certainly has changed is my father, both my mom and my dad were the first in their families to go to college. And my dad helped pay for his college through ROTC. So he would come to campus. And the one thing that really has changed is he was bringing his rifle to campus, and they shot target practice underneath the men's gym. So I don't think that would happen here anymore. Well, you can imagine if you grow up the son of two UCLA alumni, you get indoctrinated into what I call the UCLA creed. And it goes a little bit like this. I'm paraphrasing, of course. UCLA, everything about it is good and worthy. USC, everything about it's pathetic and perhaps a little bit evil. So, you can imagine when I turned 18, I wanted to rebel from this. I didn't go to USC, of course, because that I'd be excommunicated, right, from the family. So I went to Berkeley, but, it turns out the shoe's on the other foot because when my oldest decided to go to college, he went to UCLA. So that's my son in the middle. Actually, those are my two sons. My oldest son is the one graduating there, and that's my younger son. And I wanted to take the moment. I don't think there's a lot of faculty in this room, but it's funny. We have TAs in the room for sure. And it's funny that when you see students, you don't get the sense of their overall journey or their overall experience at a campus. And so I thought it was worth saying that I think UCLA is amazing. The experience he had here was just phenomenal. And so I feel very indebted to this campus. And I want to say that actually UCLA's influence on my family has not stopped yet, because this is my niece, and she's in med school here at UCLA. She's in her second year. I was hoping she'd be here today so I could embarrass her with the picture, but she has a test. Can you believe it? A med student with a test? What's up with that? Okay, so I want to talk about a little bit of science today about E. coli. And phenotype, fitness, and all these things. But I want to start a little bit by letting you know how I got into this topic in the first place, because this is entirely new to me within the last few years. As Kirk said, I worked for many years on maize. And if you don't know about maize, it really is a modern miracle. Because the thing that we eat, corn, isn't found in nature. This ear of corn, you won't find it. In fact, we didn't even know what was the ancestor to maize until the 1970s or 80s using molecular tools. So the ancestor, teosinte, has a very, very different morphology. And so I was very interested in trying to find genes that might contribute to this vast morphological shift, and using population genetics. And it seems like the methods we used were pretty quaint, but we were kind of ahead of, I'd like to think we were ahead of the game in worrying about domestication bottlenecks and measuring genetic variation and see what happens through a bottleneck, and understanding that there are actually two things happening. That is a demographic effect and selection. Okay? So we were really interested in trying to find these selected genes, thinking that they must confer these morphological shifts in maize. Well, the interesting thing is, when you look at plants and plant domestication, all kinds of stuff happens in different species. So, for example, Brassica oleracea has been domesticated and actually has led to all these different crops: cabbage, Brussels sprouts, kale, broccoli, cauliflower, kohlrabi. All of them are disgusting, and I wish no one had ever domesticated them. But nonetheless, they're there. And even when there aren't vast morphological shifts, big things happen. So, for example, in sunflowers, you start with lots of flowers, and when you domesticate it, you get bigger flowers, bigger seeds, and more oil content. It turns out, as you look across plant domestication, there are some very strong themes. That is, when a plant is domesticated, it's called the domestication syndrome. These things almost invariably happen. Larger fruits, fewer fruits, more robust plants. Loss of seed dormancy, which is an important thing if you want to grow it for a crop. So there's all these convergent sort of phenotypic effects that occur among all these domesticated plants. And it got me to thinking about adaptation. After all, domestication is a form of adaptation, right? There's selection for a domesticated crop. And it made me start to think about, what if you looked at, say, wheat and rice and maize? Are they undergoing the same sorts of changes leading to these changes, these phenotypic changes? Are the same genetic changes occurring? So in other words, this question: how diverse is the genetic response to adaptation? Are wheat and rice and maize, did they undergo the same genetic changes, or are they really quite different? And what types of mutations are beneficial? Ones that affect regulators or something else? What are the functional targets of adaptation? What are the biochemical pathways? And are there genetic constraints to evolution? That is, are there many, many ways to come up with a solution to an evolutionary problem? Or is there really strong genetic constraint? So I thought this would be really fun to compare convergent domestication, convergent evolution over domestication. And so I got together some really great population geneticists, and we wrote what I thought was a great proposal, and it got denied. And that's happening a lot these days, right? So in the meantime, I got really excited about these questions. How flexible is genetic change? If given some sort of adaptive challenge, how many ways are there to answer that challenge? So it turns out, if you're at UCI, you can't get very far from the world of experimental evolution. And I began to realize that we could use experimental evolution to address these kinds of questions. So if you'll let me be UCI-centric for a minute, since I was UCLA-centric for a while, at UCI we have a guy named Michael Rose who started experimental evolution in flies back in 1975. And what he has done, and he keeps selecting these lines for late mortality, essentially. But we also had, I think many of you know the name of Rich Lenski. He started his famous long-term experiment where he's evolving E. coli in the lab. He started in 1988 at UC Irvine. And so there's this kind of sense of let's do experimental evolution. And finally, I was convinced we should do it. But I want to tell you a little bit about Rich's experiment, because I'll be honest, all we did is copy it and do it on a bigger scale. So Rich took an E. coli B, essentially a strain of E. coli, the exact same strain, and he put it in actually 12 different tubes. So he had 12 replicated experiments. Every day he does a 1/100 dilution, and so on and so forth. Now in that dilution, you think, wow, that's a bottleneck. That's really making a small size of the population. Actually, the populations are still huge, on the order of 10 to the 6th to 10 to the 8th. So there's not really much population bottlenecking, and these are very large populations where selection is very efficacious. So what is the selection? In this case, he took this E. coli B that's very happy at 37 degrees, and he put it in a glucose-limited media. So the selection was to grow well in the lack of really many nutrients. So they've been doing that experience since 1988. Every day someone goes into the lab and does serial dilutions of their populations. The really clever thing that Rich did is he figured out a way to measure the fitness of the evolved lines in a very simple way. So what he does is he has an ancestor, and then he has the evolved lines, these lines that he's evolving every day, that have a color marker. So it's actually very easy to mix them together at time t equals 0, and then let them grow together to see which one is outcompeting the other. And in this way, you get a sense of the relative fitness of the evolved clone compared to the ancestor clone. And it really is as simple as counting different colored colonies on these bacterial plates. So you use a very simple equation, and you get a sense of relative fitness. And here's what Rich found many years ago. And that is that you start, of course, with a relative fitness of 1, because nothing has evolved yet, and that the fitness goes up very rapidly and then starts to saturate. And it saturates within a couple of thousand generations. So Rich did this very nice study, 12 replicates. And we decided we would like to do that. We being two great colleagues, Tony Long, he and I were hired the same day at UC Irvine. I don't know if you know Tony, but he's fantastic. And Al Bennett, who had been at Irvine for a long time. And Al was always interested in temperature. So temperature, physiology of lizards, believe it or not. But with Rich Lenski, he wanted to see how bacteria responded to temperature. So we have this kind of history of looking at bacteria. And we decided that we wanted to then ask these questions about how diverse is adaptation. And we did it in the exact same system as Rich Lenski, but with a few small changes. First, we started with one of his lines, Rich Lenski, that was adapted to this low glucose media. And then we grew them at 42 degrees. Now, why 42 degrees? This is about the limit at which they're happy. After that, the populations tend to die. Okay? And then we did the exact same thing that Rich did. And so you might call this sort of a bonehead experiment, except we did it on a much larger scale with 115 replicates, and we did it for a full year, 2,000 generations. And at the end of this experiment, we took a clone, and we did, sort of the obvious things, such as we measured the relative fitness. And at the end, after 2,000 generations, the evolved bacteria after a year were 40% better than the ancestor, growing at 42 degrees. So a remarkable change. By the way, I wanted to say one of the things about using temperature, this was purposeful, is that there's complex ways to adapt to temperature. It's not like an antibiotic where you might have one gene that might be able to mutate and then it evolves resistance. Temperature affects all the cellular processes, and we purposefully wanted sort of a complex response to whatever selective regime we gave these things. Okay, so after a year, they're about 40% better. And then, of course, we sequenced each of the genomes of these 115. And here's where someone very important, an old friend of mine, Olivier Tenayon, was doing a sabbatical in my lab, and this would not have been as cool without him. So we sequenced all of these genomes, and here's sort of a schematic of the genomes. And you can look at it, and there's different colors representing different kinds of mutation events, point mutations, insertions, deletions. And if you're interested in diversity, right, you can look at it and say, so what's in common? This looks to be in common. So that actually was found in over 35 of our lines, and that is a deletion that's actually mediated by transposable elements on either side. And so we think there's a very high mutation rate. They recombine, and then you get a deletion, and it's probably very advantageous. That's why we saw it in so many lines. But you can see otherwise, it's a little bit hard to pick out patterns. And I'm going to talk a little more about what we found in there in a minute. In total, we found, 1,300 mutations. And you can see most of them were point mutations. That's the bar on the left. But we also saw small insertion deletions and all kinds of other things, not surprising. On average, there are about 11 mutations per sequence. And most of the mutations, I want to stress, we think were beneficial. You can use things like measuring the ratio of non-synonymous to synonymous mutations. If it's greater than 1, suggests it's beneficial. But also remember, we set up the experiments so there are big population sizes. So there's not much genetic drift, efficacious natural selection. OK, so how replicable is evolution? This is one of the things I wanted to answer, right? How diverse is the response to an adaptation? So you can measure replicability across our 115 lines or so by different ways, by just looking at mutations, or by saying which genes were mutated, or you can even, the nice thing about E. coli is a lot of known about protein complexes. So you could say, how often is a particular protein complex mutated? And what you find is that on average, 2 lines share about 2.6% of their mutations. Right? That's very low. On the other hand, so that suggests there's a lot of variation in the way evolution responds. But there are some things that are found quite often. For example, in this gene RNA polymerase, which I'll talk a lot about more, there's 18 lines that have a mutation in the same codon. And this other thing, which I'll again talk about a lot more, the rho termination factor, 17 of our 115 lines had a point mutation in the same codon. So there's a lot of variation, but there are some themes. You can find things like this when you look at the actual gene level. How often is a gene targeted? This one gene was mutated in 65 lines. It wasn't always the same mutation. They were all knockouts, though. Right? So clearly, knocking this gene out was something worth doing in terms of the fitness at high temperature. So one of the nice things about our data set was it's big enough that you can play games like this one. And that's if you want to ask, how many targets of selection, how many ways to respond to high temperature? You can resample the data. Okay? So let me show you what we did here. Here at the bottom, we resampled, maybe here 20 out of our 115, 40 out of our 115, 60 out of our 115, and did that 1,000 times. Okay? And then you'd ask, for example, this line, how many genes do you find that are targeted, that you find mutations in when you do this resampling? Okay? So how many genes do you find? You see we quickly saturate at about 60 genes. Okay? So that tells us 60 genes or so seem to be important in the response, this adaptive response to thermal stress. Okay, that's cool. But what's really, I think, kind of blows my mind still, this is an old paper, but I have to tell you about it to tell you about the new stuff. What blows my mind is that when you look at point mutations or just any kinds of mutations, we're not even close to saturating. So what does that mean? What that's telling us is that there are a limited number of genes that seem to be important for this response to thermal stress. But within those genes, there is, at this point, an unestimable number of potential mutations that can contribute to adaptation. Now, I think that's crazy, because if you'd asked me beforehand, I'd say, OK, you can take a gene and it has an active site. And maybe you can change 1 or 2 bases in that active site and it's going to change it. But those are the only things that matter. That's not the picture we're getting here. We're getting a picture that there are lots of potential changes that can be adaptive. How many potential changes? I don't want to go into the details, but what we did is we did this thing called the coupon collectors problem. It's a pretty simple idea. The idea is this. You want to collect coupons. And you know that some company is putting out 800 coupons. How many times do you have to go to the store to collect all 800 coupons, right? And that's a probabilistic kind of issue. You're not always going to get a new coupon when you go to the store. On the other hand, we did this. We found our most certainly beneficial mutations because they were found in more than one line. We said, we have collected 400 coupons already. We know we've sampled 115 times, so how many beneficial mutations are possible? And you can quickly get from an estimate twice our observed number up to 850 under very simple assumptions. And with just letting the assumptions go a little bit, you can estimate the thousands and thousands of potentially beneficial mutations in the genome. So the main point was, we didn't do enough replicates in some ways. It's the biggest experiment that's been done at this time, or at least at that time, and we could have done more. So the other fun thing about it is now you get all these data of 115 genomes, and you can do things like make the square. And we're going to look at the square a little bit, so I'm going to make sure you understand what's going on here. So, for example, I told you about this gene, YBAL. Every time there's a square here, say this is another gene, CLS, every time you see a circle in there, it means you had mutations in one genome in both of those genes, YBAL and CLS. Okay? So every dot that you see means there's a co-occurring mutation within one genome. Okay, so if you look at that square, you can say, okay, wait, there's something weird here. The first thing is there's some empty squares. Okay, so for example, YBAL was never mutated twice in any of our experiments. Well, that makes great sense. Why? Because YBAL was always being knocked out. Once you knock it out once, there's no point in knocking it out again. Or if there is a mutation that knocks it out, it's not strongly selected. It's not advantageous. So that is, by definition, negative epistasis, essentially, right? So by looking at the square, we can learn about epistatic interactions. That's my point. So we also found other things. YBAL makes perfect sense. But look at this circle here. You can see it's called ROD. This is a series of 6 genes, all of which were found to be mutated in at least one line. But never did we find mutations in more than one of those genes. And probabilistically, that's not expected. So for some reason, there's some sort of, it seems like, negative epistasis. Once you get mutated in one of these, you don't get mutated in the others. What these genes are doing for thermal stress, I have no idea. Absolutely no idea. So then there's this one down the diagonal, RNA polymerase. Interestingly enough, it is the only gene that had more than one mutation in it within a single genome. RNA polymerase, of course, is absolutely critical, so it's never knocked out. And it was also the most mutated gene in our experiments. So it's highly pleiotropic. Of course, if you mess up RNA polymerase, you're messing up every gene that's transcribed in the genome. So one thing isn't clear is why does it get more than 2 mutations? And we sort of have a pet hypothesis that once you get one mutation, it affects a lot of things, and then you get another one, it maybe compensates somewhat. We have some evidence to suggest that's true, and I'll get to that in a little bit. So in the end, what we have is the square, and this is actually taken from a Nature Reviews Genetics. This is our data, though. What we have is this really interesting sort of picture where you can color the squares according to the darker, the warmer colors represent, you tend to get mutations that associate positively, right? So these squares, it tends to be that when you get a mutation in ROD, you also see a mutation in RPOB. Okay? So you have this positive these positive interactions, and then these cooler colors are negative interactions. And what we found by using this approach is there's actually 2 ways to evolve to thermal stress. And in fact, they're not mutually exclusive, but they're probabilistically, you tend to choose one path or the other. Either you mutate your RNA polymerase gene, or you mutate this Rho gene that terminates RNA polymerase. Both of these pathways affect transcription. Okay, so we have 2 paths to adaptation. And that, of course, leads to a lot of different questions. Now, just to be sure, I think everyone knows what RNA polymerase is, but one of our pathways was RNA polymerase. With these ROD genes, how they interact, why they interact, we have no idea. The other one is this termination factor Rho. And it is, I think, the coolest molecule ever. Because what it does is it actually sits like a circle around the RNA as it's being transcribed. And it goes down the RNA and then runs into the polymerase and knocks it off. right? So not every gene is terminated by Rho, so it's not affecting as many genes as RNA polymerase. But for some reason, these two things seem to be separate, exclusive, somewhat exclusive pathways to evolve thermal stress. So up to this point, I thought, wow, it's our first ever E. coli paper, got published in Science, right? Life is good. This is so easy. Turns out it's not so easy. It actually gets really hard after this. So we're slogging our way through lots of things, like questions like, both of these pathways modify transcription. What's the mechanism? I'm not an E. coli biologist. It's hard. Why are they negatively epistatic? If you take one path, you tend to not take the other path. We have some ideas. We don't really know. Do these two pathways differ in fitness and phenotype? That is, are you in better shape if you go one pathway than the other pathway? And what is the speed and direction of adaptation? And I'll talk about what I mean by speed and direction in a minute. Okay. So we've actually been focusing on these two bottom questions now, and I want to tell you just a little bit. It's sort of the tip of the iceberg, but some of the things that we're finding. So I'll start with this question. Do the two pathways differ in fitness and phenotype? I'll tell you one thing. Remember, we can measure relative fitness. We can measure it at 42 degrees. Among all the evolved clones, there's no difference whether you take one pathway or the other at 42 degrees. But there are differences, so I'm going to tell you about some differences we found. So I had a really great graduate student, Alejandra. She's taken off for Switzerland, where life is better, the chocolate's good, and she was really interested in thermal trade-offs. Okay, so the idea is following. You have an organism, it's E. coli in this case, that grows very nicely at 37, that's its optimum. And then it has some temperature niche. It has a range of temperatures at which it grows. And when we did our experiment, we were telling it to grow at the very edge of its niche, 42 degrees. So the question then is, what happens to the niche? Does it shift entirely so that you, at lower temperatures now, you're less fit? Or do you just expand your niche? Or do you specialize your niche? These are kind of interesting questions about trade-offs. Is there a trade-off at low temperatures? So what she did to look at low temperatures and high temperatures, the borders of these temperature niches, is she grew her cells for 4 days. And she did serial transfer, and all she wanted to know is, can bacteria reproduce itself? Okay? In this case, she would take a slope. The slope is 0, and we're going to call that the absolute fitness. And that's when a population can persist. But if it's at a temperature that it really doesn't like, it's going to do this. Over time, the population crashes. You get a negative slope. And then we say that it doesn't persist. So if you do this at 43 degrees, and I don't want to get into the details of this, but you can see the evolved lines are super happy. They just grow. Their slope is 0. The ancestor crashes. How much it crashes depends on how often you look at it. But in general, it sort of crashes, kind of persists, and then crashes again. So the upper limit is 43 degrees for the ancestor. And for the evolved lines, they grow to at least 45, but we don't know how much farther they'll go. The really interesting thing was at 17 degrees, or at lower temperatures, and this is supposed to be moved over. Sorry about that. This is to show you that at 17 degrees, all of the populations, including the ancestor, simply don't grow. So over time, the population declines. But interestingly, at 18 degrees, you get this really weird dichotomy that half of them grow and half of them don't. That's kind of cool. So, I don't want to ask that question yet. What I want to do is tell you a little bit more about what happens at low temperatures. So again, I told you 52% don't grow at 17 degrees. You can't see this now, I'm sorry. This is 18 degrees. So they grow fine at 18 degrees, and they behave just like the ancestor, which is the dark line, in terms of growing at low temperature. At 35% of the lines don't grow at 18 degrees, but do grow at 19 degrees. And then we have the remainder that don't even grow at 19 degrees. In fact, some of them don't even grow at 20 degrees. So in terms of their shift in niche, what's happened with these is they're still fine at low temperatures, but now grow much better at high temperatures. These guys here have just shifted their whole niche by a couple of degrees. And these guys have actually narrowed their niche because their low temperature ability is really pretty bad. So then the question becomes, what's the genetic causes for these differences? And as you can imagine, we're going to get back to Rho versus RNA polymerase, because that's where the story ends up. So we did, to start with, just genetic association, an observational association. For anybody who's done associations, we have absolutely the worst data in the world to do this. Because when you do associations, you want things to be at a sort of a reasonable population frequency. And almost all of our mutations would be here where we haven't plotted them. They're single. You see them in only one genome. Okay? So these are horrible data for doing associations. But we have a few mutations that are at relatively high frequency, 15 out of 115 lines, for example. And you'll see that a couple of those include mutations within Rho and RPOB. So if you do associations, what you find is, oh, sorry, that's just some details. What you find is if you look at all the clones that have this one mutation in RNA polymerase, it's only one of many mutations in RNA polymerase, but there is a strong association. They just can't grow well at low temperatures. This is 18 degrees. You'll see some of them in blue do actually grow reasonably well, and I'll come back to that in a minute. On the other hand, if you look at all the lines that have this one mutation in Rho, they grow very well at low temperatures, except for this one. So what's going on with that one, we don't know. Here's what's interesting. The ones that actually grow well at 18 degrees actually have a second mutation in RNA polymerase. So it does suggest there's some sort of compensation going on. But why? Why would there be compensation? We never grew them at 18 degrees C. You got to remember that. We're showing them this is something they've never seen before. So what this is actually doing, what the compensation is at high temperature, we don't know. Right? Okay. So it turns out, now that we kind of got this data, if you just take all of our lines and say these are the ones that mutated in Rho and these are the ones that mutated in RPOB, there is big differences at low temperatures. So that's one of the things about using two different pathways, that there's certainly a trade-off difference in terms of low temperature growth. Now remember, we grew these at high temperature and at constant temperature. I would predict, I don't know if it's true, it should be easy to do, if you grew them at high temperature and then cold and then high and cold, we would only find the Rho pathway working, because it can grow at both temperatures. But it's interesting how you can have these different ways to act genetically, all to the same stress, but they have very different outcomes in terms of their sort of periphery effects. So the nice thing about E. coli is you can actually test what you think is happening by making mutations. This is why I love it. It's hard to do in plants, but you can do it in E. coli. So we made the single mutations just to make sure. So we took the ancestor and put that one mutation in there, Rho 115N. And what happens? Well, it grows just as well as the ancestor at low temperatures. If you do this for a bunch of different RNA polymerase mutations, so we did 4, you find they always grow poorly at low temperatures. They're always worse than the ancestor. Okay. So what did we learn from this little part of the talk? We learned temperature and fitness trade-offs are common. Not always, though. That these two pathways that are solutions to thermal stress actually differ with respect to trade-offs at lower temperatures, but they don't differ in fitness at 42 degrees. So trade-offs, we know, are in this case, are generated by antagonistic pleiotropy. That means our RPOB mutations are clearly very advantageous at high temperatures, but there's a fitness trade-off. It's an antagonistic pleiotropic trade-off at low temperatures. And what are the mechanisms? Why is one affecting low temperature growth and the other isn't? We don't know, actually. But we think what's happening is RNA polymerase, if you mutate it, you affect every gene, the expression of every gene. But if you mutate Rho, you're affecting the expression of a subset of genes. So we think there's just more pleiotropic effects. You're affecting more sorts of traits when you mutate RNA polymerase. Why are they in negative epistasis? Again, we don't know. We're kind of trying to work on this problem, but we think this is what happens. When something is growing at high temperature, RNA polymerase, and this has been shown, increases its rate. Okay? So we think the mutations are actually slowing down the rate of transcription, the mutations in RNA polymerase. We think also the mutations in Rho are also affecting termination. Wait, let me say this again. You slow down RNA polymerase because it has to slow down to terminate well. We think Rho is also doing this, and when you have both of them together, boom, too much termination, you're not a healthy organism. We don't know that for a fact yet. So, how much time do I have?
2 minutes.
Oh, yeah, I'll be done, no problem. So, we learned something about these two different pathways do have different phenotypes. What about the direction of adaptation? Now, let me explain to you what I mean. So in our experiment, we take this very happy 37-degree E. coli, right? It grows happily at 37 degrees, and we put it at 42 degrees. And when it does that, it's under physiological stress, and it has to acclimate, right? So before there are any genetic mutations or any mutations that have driven to high frequency in the population, there's this physiological response. And then after that, we go through 2,000 generations where there are genetic mutations and there is actually genetic adaptation, right? So that's the adaptation part of the story. So the question is, does adaptation, does it really just sort of try to reset physiology back to 37 degrees, right? So adaptation is just trying to calm down this organism that's not very happy at 42 degrees. So that's what we call restoring. Does it restore the pre-stress phenotypes in physiology? Yeah, sure.
At the end,
Yeah.
do you see the evolved bacteria are sicker compared to the ancestral bacteria?
Are what?
Are they sicker? For example, if you look at the growth curve, does it take longer to...
Yeah, I'll show you that in just a minute, actually. Yeah, it's a great question. So I'll get to it, I promise.
Okay.
We're also interested in the speed of adaptation. So how fast does it happen? Remember, when we look at Lenski's curves, adaptation happens really fast, within a few hundred generations. Okay, so we're going to look at direction and speed. And we're going to do that by looking at gene expression. And we also are interested, I should say, in what happens at the very beginning, large effect mutations. And it turns out we know something about these first step mutations because they are in, not surprisingly, RNA polymerase. And for some of these, we know this is a graph of how quickly they rose to fixation in our experiment. Some of them fixed really rapidly within a couple of hundred generations. So these are what we're going to call first step mutations. So now we have this experiment, 37 to 42, physiology. The first step, genetic adaptation, and then adaptation to the end at 2,000 generations. Okay. And then we're going to look at genotype at all these 4 different states. Okay, so let's look at this first comparison, 37 to 42, and we're going to look at RNA expression data and at growth curves. So here we go. I promised, right? Okay, so here's the ancestor. It grows very nicely. It has a nice log exponential scale. When you put the ancestor at 42 degrees, it's just not a happy camper. It takes much longer to get out of stationary phase, and it just never really gets going, and its yield in the end is not so great. So growth is slowed and yield is down. Now what you can do is, to the extent there is an exponential phase here, you can get RNA-seq data from both sets of populations, from mid-exponential phase. And this is, again, Alejandra's work. This is unpublished. And I'll tell you why as we go along. So you can do the classic RNA-seq types of things and see how many genes are expressed more highly in the ancestor, how many are at 37 degrees, how many are expressed more highly at 42 degrees. And you find that there's about, how many genes? 2,000 genes that are differentially expressed, right? So this is a very big response between 37 and 42 degrees. So roughly 50% go up and 50% go down when they're under stress, 42 degrees. And remember, this is a physiological, not a genetic response, right? We haven't had any adaptation yet. This is just letting the poor bacteria sit there for a day and be really, really unhappy. Okay, so what happens when you have one of these first mutations in RNA polymerase? with just the single mutation that we've engineered into the ancestor, you can see that growth looks much happier, right? They grow, they come out of stationary phase a little bit earlier, but then their exponential growth phase is really nice and they get a much higher density. Okay? And what you can do is compare gene expression in this guy, our first mutation, versus this guy, our stressed out guy, and you find there's a bunch of changes around 1,600 different genes. Okay? So now we want to know, what's happened with these 1,600? Are they being expressed in a new way because we have adaptation? Or are they actually just being expressed as the organism likes it at its optimum of 37 degrees? So to do that, we did sort of a funky plot, which measures, we have on one axis the difference in expression between the two temperatures for the ancestor. On the other axis, we have the ancestor at one temperature versus the major mutation. There's a couple of weird things about this plot, but the main thing I want you to see is that there's this really strong negative correlation. And essentially, that negative correlation is telling us that once we get that first step mutation, a lot of the genes are simply being reverted back to its gene expression state at 37 degrees. So we go through physiological stress, gene expression goes nuts, you get this first step mutation, RNA polymerase, which affects a lot of things, and suddenly lots of things are going back the other way. Now this is a weird correlation because there's a built-in negative bias to it. I won't go into the details, we need to do some work on it, but any way you look at the data, you see that sort of effect. If we look at another first step mutation, the other one was phenylalanine, this time we have a leucine, you see the same kind of thing. Similar numbers of genes being affected, and a similar correlation. Gene expression is going back to the optimal state. Okay, so then what happens? We've had a first step mutation, and then we get to the end of the experiment, which is 2,000 generations. And this is getting fixed, 100 or 200 generations, right? So there's a big difference in between these. And what's happening there? Here's the growth curves. See, I gave you too many growth curves. I'm sorry about that. But this puts them all together. Here's the ancestor when it's super happy. Here's when it's super sad. Here's when you add one mutation. It grows much, 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 much better. You can see that. And then through the next 1,800 generations or so, it does even better. Comes out of stationary phase faster, grows better, higher yields, right? OK, so clearly something is happening from that first mutation towards the end. And what is it? Well, when we look at gene expression data, we don't know what it is. It's something very subtle. We can't pick it up. This is all nicely replicated data. It's as good as you can get it. So these are two different lines from the end of the experiment that have the mutations that we've assayed here. This one, you can see there's 51 differentially expressed genes, but most of these are actually, let's be honest, they're background level. They were deletions. The genes aren't even there, right? So you can't really say they're differentially expressed. This one, we have 10 differentially expressed genes. So we're seeing big changes in growth dynamics, but very few changes in gene expression through the latter part of the experiment. It's a mystery. We don't know why. OK. So at the first part of the experiment, we have 2,000 genes or so that are differentially expressed. Those 2,000 genes, again, are differentially expressed after the first mutation. And at the end, we're seeing less than 100 genes, though they're clearly affecting fitness in the growth curve. So it's very interesting. I forgot what I wanted to say here. OK. So it's clear this major effect, these first mutations have a big effect, right? They're restoring gene expression. So what's up? Is this really the way that evolution works? After all, we're looking, let's face it, we're looking at a silly little experiment in the lab with E. coli. It's not very realistic. So are we learning anything about the greater world, about how evolution works? Well, maybe. I say that because whenever you look at evolutionary experiments in the lab, it seems RNA polymerase is almost always mutated. So it seems that this is a way, at least in the lab, under simple lab conditions, that things happen. But then I can also go back to my plant domestication roots. And we actually know quite a few genes that have affected domestication. And over half of them regulate transcription. So I don't want to conclude this fully, but there is some evidence to suggest that lots of the first step big mutations might in fact affect transcription. And there's old ideas about that, actually. But in terms of evolutionary theory, if we are really far from the optimum, if we're starting our populations and pushing them towards an optimum that's very far, sort of in the fitness state, you expect very big affect mutations. So maybe the fact that these mutations affect things so much is really just a function of our experimental design. And there is one other big but. It turns out that doing RNA-seq data in E. coli is really hard. You'd think it's really easy, but it's not. Because if you capture the cells at the wrong part of their cell cycle, you could have real problems. So we are nonetheless repeating especially this point here. Because if we happen to catch that wrong point, maybe what we're learning is all BS, and so I'm sorry I told you anything about it. So let me conclude everything that I've said today. Boy, I've been talking a long time. We found some insight into the flexibility of adaptation. I want you to go home with the idea in this simple experiment where we're evolving things to 42 degrees, there's probably 60 genes that play a role and an unlimited or unestimable number of sites. So evolution is remarkably flexible in terms of genetic adaptation. We found two major adaptive pathways. In part, they seem to be governed by epistatic interactions. If you go one way, you don't go the other. The two pathways do not differ in fitness at 42 degrees, but they do differ at low temperatures. We can show that some of these mutations really generate antagonistic pleiotropy. Adaptation seems, we're not 100% sure of this, to mostly restore expression towards patterns at 37 degrees. So it's just the idea that at 42 degrees these things are freaked out, and the best genetic changes are the ones that make it behave more like it was at 37 degrees. Most of this restoration appears to be achieved by single large effect mutations. And that's what we were showing with these mutations in RNA polymerase. But we do have data, I will add, in Rho that suggests it's not the same story. So we're trying to figure that out. I'm going to skip this and just acknowledge all the people who've been so wonderful to do this stuff. Olivier and Tony and Al, I've talked about. Alejandra, Sean's a grad student. Becky is not only my wife, but my technician. And I don't know if it's better to say I married her before she was my technician or she was my technician, but I married her before she was my technician. Pam's another technician, David's a statistician, and Andrea. And we've been, the UC Nexus Fund has been so amazing for this group because it's supported Alejandra, David, and Andrea in their studies, so it's been fantastic. So that's it, thank you. Happy to take questions. Yes?
So within E. coli, do you think that, in a sense, or can you speculate, do you think that you would see a similar set of genes being mutated if you were trying to get them to adapt to, say, 40 degrees versus 44 degrees?
Yeah, it would be complete and utter speculation if you allow me that. I think if you're going to a stressful high temp, it's going to be about the same sets of genes. That's my guess.
So what
Yeah, maybe you don't need as remarkable shifts. It's not entirely clear to me. I mean, there's so many things you could do after this experiment, right? And that's one of them. Another thing I'd really like to know is how far can you push them? I mean, can they become, thermophiles, right? They're growing at 60 degrees. I have no idea. We don't know that. That's a good question, though. Thanks. Yes?
Very interesting talk. I guess
Thank you.
So many directions. One direction I see is I've noticed the U shape in when you grow them in low temperature, 17, 18C, right? Sometimes there seems to be evolutionary rescue.
Yes.
It's going down and up. I think it's a very interesting topic, and maybe playing with the population size, then you might be able to say something about the likelihood of rescue.
Yeah. So actually, we don't see rescue at low temperatures. We definitely see rescue at high temperatures, and that's what I said.
I do see one or two.
Oh, you did? Okay. Yeah, it may be the way it's graphed, but when you look at the data carefully, we really don't see it. But at high temperatures, it's amazing. So you can, the ancestor dies at 43 pretty consistently, but at 45, it actually recovers. So it goes bam. And so it's getting these mutations really fast. So we're actually trying to look at the relationship between sort of the frequency of rescue to temperature, and also what is the rescue? My bet would be it's almost always RNA polymerase genes, but who knows, right? Yeah, it's cool.
Question,
Sure.
Maybe not suggestion is,
Yeah.
I think another interesting question is reversibility. So people talk about how reversible adaptation is given the complexity, right?
Yeah.
I guess when you say first step and then to the eventual adaptation, I would assume it's all the small effect beneficial mutations accumulate over time.
Yeah.
Will make it less reversible. If you actually grow the 42 adapted strains into 37 and then
Yeah.
see what happens.
Haven't done it yet. I'd love to do it. It's you know, these experiments, I never knew why they were. When we wrote this, so I didn't know anything about E. coli. I would still argue I really don't. But they take a lot of manpower, surprising amount. And my poor wife, for a whole year, she had to go in every weekend and every holiday. Needless to say, it was a horrible year for me domestically. Yeah. But yeah, I'd love to do it. It's a great idea. Thank you.
Or at Harvard, they have this automated.
They do the Desai lab, yeah. E. coli is much, yeah, I've never grown yeast, but E. coli is it's harder to do in the high throughput way. Yeah. Yeah. Other questions?
Do you see constraint on the sequential steps of how the mutation occur? Like, for example, like, does certain mutation have to happen first or another set of mutations to occur? Or like, is there any initial mutations that determine which of those two passes?
We don't know. So one of the things, I skipped a slide about some of the things they'd like to do, but we're hoping sequencing costs have come down enough, especially library costs. It's not so much the sequencing costs, but the library costs, that we're hoping we can sequence every time we saved every 200 generations or something for every line, to ask exactly those kinds of questions about contingency and issues like that. Yeah. Yeah. Just, we don't have the data. Vicky.
So how much, you've got at least two different pathways in this very simple experiment.
Yeah.
Which suggests in nature, we're likely to get lots of pathways. So how much is the genetic context of the population shaping which of the pathways? In other words, the genetic environment of your different lines, is there any way to pick up whether or not that it was more likely to shape it to go into this mutation pathway versus that?
Yeah, so I can tell you this. If we take those same RNA polymerase mutations, the ones in codon 572, and we put them into K12, which is a very different background from E. coli B, they're not advantageous at 42 degrees. Yeah. So I think, again, that's underlying the simplicity of this system. I thought we learned a lot, but in the end, we can say, how much does this apply, right, when you have different genetic backgrounds? Yeah.
Exactly. Genetic backgrounds, which is what you're going to get in nature. It would suggest that there's going to be lots of ways to deal with high temperatures.
Another way to say this is we're underestimating flexibility in this experiment, right? Because what you're arguing is if we start with more genetic backgrounds, there are going to be more pathways. Yeah, and I completely agree with that. I'm not sure everyone would.
Well, this is what was suggested by this work.
Yeah, no, I agree. Any other questions? I do want to say, we do have two members of the class of 57 sitting in the back, my parents. Sorry to point you out like that. And my brother as well. Turns out they live in Westwood, so I've spent a lot of time in Westwood over the years. Any more questions? Thank you very much.